Our focus will be on what is referred to as the central dogma of biology, protein synthesis. I oftentimes see students that really the universal answer in biology is proteins. And so the focus is going to be how proteins are manufactured. We do know that the information is stored on DNA, but how that gets processed into a usable protein by cells is our focus. So as we move into that, then what I would like to do is start with structures referred to as RNA. RNA is another genetic um, piece of cells. There are two types of genetic information. One is DNA, it's that double helix, and the second is RNA. Its the full name is ribonucleic acid. There are, just like DNA, there are bases that correspond with RNA, and we again call those nitrogen bases based on the nitrogen that's present within those. The four that are present are cytosine, guanine, adenine, and a different one this time, and that's to make note, and that is uracil. So uracil down there is the fourth. That's a little bit different than the DNA. The other thing that you see that's different is that RNA has not only these bases, but it has the sugars and the phosphates here in red. But it only has one side of it. So there's not two sides. So it's definitely not helical as far as with a complementary side, but it is a single sided molecule with phosphates and sugars for sure, and also some of these bases. So there are some major differences, and we alluded to those in the last slide. The first one is that RNA is single sided, whereas DNA is double sided. The second is uracil replaces thymine. And the third that you may not pick up on as clearly, but it comes in the name, is that the sugar that's on the side over here, so you have the sugar phosphates here. In DNA, this sugar is deoxyribose. That's the name of the sugar. In RNA over here, the sugar in red, and it's a hexagon ring, is a sugar called ribose. So those are the three main differences between DNA and RNA. And that's what we're going to focus in on as far as how that's assembled and how it's used within protein synthesis. So as I mentioned, um, RNA is a little bit different, but there are different types of RNA. Number one, there is one called messenger RNA. Number two, there's something called tRNA. And number three is rRNA. So all these have a lowercase letter and then uppercase RNA. So the M, T, and R are three different types of RNA. So let's focus on the first one, messenger RNA. Messenger RNA has the capacity to carry a message from its location on the DNA molecule, which is inside of the nucleus, to the outside of the nucleus in the cytoplasm, where there is something we refer to as the ribosomes. And ribosomes have the ability to build proteins. So we're going to assemble the message, and then we're going to bring it out to the location of the protein, or to the structure that manufactures it. So that's number one, messenger RNA. Number two is something called transfer RNA. Now, this looks really complex. If you look down over here, you'll notice that the yellow arrow is indicating something referred to as an amino acid attachment site. Now, this is very, very important. One of the things that tRNA does is it carries amino acids to a location in the cytoplasm to the ribosome. So it's a transfer. It's transferring, in this case, an important cargo, and that cargo is something called an amino acid. So that's number two. The only thing that I guess I would like to do or suggest before we go to the next slide is that it has the shape of a T in some ways. Up on the top here, there is an area where an amino acid could attach, and then the rest of it has a D stem and another TC loop is called, and then a variable site down below. But each of these areas here represents one of the four bases: adenines, guanines, cytosines, and uracil. So that's what you're seeing all the way around here. It's just kind of shaped differently. The third one is actually another name for the ribosome, and that is rRNA. So rRNA is actually where everything is coming together. Messenger RNA is coming to it, and tRNA is transferring substances to it. So it's the factory where all these things are going to come together. And in other words, all three types of RNA are going to assemble themselves on the ribosomes. So the purpose of the ribosome from earlier years that you may have remembered is where proteins are manufactured, the site of protein synthesis. That's the job of the ribosome. Well, the ribosome is actually a type of genetic material. It's rRNA, and it has the ability to attach a messenger RNA temporarily and also bring towards it as an attractive site the tRNAs, which carry the amino acids. All right. So, having stated the three types of RNA and some of the differences, let's get at the crux of the matter. The crux of the matter is this idea called protein synthesis. So, in other words, how proteins are manufactured. The diagram here that you can see here is my double-stranded molecule of DNA. And as the terms are describing it, there is a term that describes the first part of this process is called transcription. And then there's an arrow indicating a type of RNA. This is a type of messenger RNA that will transfer a message from this molecule, the DNA, out to the ribosomes where a protein can be eventually manufactured and produced. This idea is oftentimes referred to as the central dogma of biology. In other words, it's the big idea. That's why I tease students that really, if you're on a multiple choice test, if you're unsure about the answer and one of the choices is proteins, you always choose it. Now I'm kidding, but proteins are the functionality of and the information area is found in the DNA, and it has the instructions of how to build proteins. So here's the two parts of proteins from that diagram from the previous slide. The general overview is this. The first part is called transcription, where DNA is unwound, and a segment of it is copied. So I think that the start with is I'm just going to highlight here with a pen, for example. If I were to unravel a piece of DNA, but not all of it has to be unraveled. So let's just say this area right here, it has the information for my favorite gene, and that's nasal hairs. So we'll just label that as big H. So the first thing that's got to happen here is that we have to unzip, or what has to happen with transcription is transcription has to unzip this section of the DNA, and when it does so, it'll begin to assemble a messenger, which we refer to as messenger RNA. The second part of that is once we have the RNA, or in this case, the messenger, we're going to bring it out to the ribosome and begin to manufacture the protein. This is called translation. So there are two parts, transcription and translation. I always think of transcription as like a physical copy. So if I go down to the counseling office and I ask for a transcript, they're going to give me a physical copy of all my grades here at Hopkins. And then you can bring that transcript home and you can translate that to your parents or other people saying, well, what does a B in honors biology mean? Well, a B in honors biology, Harris is a really tough grader. He's got long nasal hairs, and you can explain to them why a B is a really good grade. But that's the translation part. So two parts of protein synthesis, transcription, a physical copy, which is in this case messenger RNA, and we're looking at the second part, which is translation, which is the physical protein that's going to be eventually manufactured. All right, let's see if I can move ahead here without causing too much challenges here. We'll erase all that now. All right, let's move on to the next slide. Hopefully. Not yet. So the first part of this is called transcription, and I apologize just down below that this is an N. So transcription is where the DNA message is copied, making what we refer to as messenger RNA. So let's take a look at that. The first part that's going to be necessary here is an enzyme called RNA polymerase will be released onto and attached to the DNA molecule. Once this happens, what you're going to notice here is that the DNA will unravel. Remember, it's a helical structure. So RNA polymerase attaches to the DNA molecule and it unravels it. So what happens here is we have these bases that are exposed. And so you can see the two purple bases here, the green one and the red one and another red one here. Those bases are my typical nitrogenous bases, adenine, guanine, cytosines, and thymines. And then number two, when this happens, it gives it an opportunity for some of these free-floating nucleotides that are present that can be potentially complements to that one side. And this is the process or the beginning of this, and where you can see at this point, um, this guanine in purple will attach to a free-floating nucleotide. In this case, the matching up is cytosine, and this one over here is guanine. This one would be the green one would be cytosine. And then what you're noticing here, this red, this thymine is going to attach to an adenine and so on and so forth. Remember that there's a base called uracil. So for example, this adenine will only bond with a uracil, not a thymine, on messenger RNA. So what you're beginning to see is this assembly of this molecule that's copying one side, not both, just one side of the DNA molecule. And when that happens, you can see this long chain being copied. There is a starting point and then there's an ending point. So this polymerase continues until it reaches what is referred to as a terminal site. So in other words, this is like the period at the end of a sentence. Once it reaches this point, it gives an indication that the copying of that information, that gene, is complete. And now what will happen, messenger RNA will be released from this molecule, and DNA will coil up and go back into its original double helix. So there's this genetic code, and this code on messenger RNA, we give it a special name. It comes in groups of three. It's almost like a special code that has three letters to make one word. So, for example, AUG is the first three letters. We call these first three letters a codon. Right next to it is the next group of three letters, almost like another word, that is also called a codon. So on messenger RNA, remember that there is, in place of a thymine, there's a uracil, and it comes in groups of three. So pretend like this is a sentence. AUG is a word. Another word is right here, ACA. And these are codings that we're going to interpret out in the cytoplasm with the help of tRNA and the ribosomal RNA. But right now, we just made a copy of that information in groups of three from the DNA molecule. So this messenger RNA strand will go out and begin part two. Part two is something called translation. So after it leaves the nucleus, the messenger RNA travels to the ribosome and it will attach to the ribosome. So you can see my messenger RNA right here. The green area is my ribosome, and it's going to read this message, this codon sequence. And so you can imagine here is my AUG, which is like a capital letter. It's the start sequence, which we call a start codon. And then it continues on in groups of three: UGC, AUA, CGC. And again, it's doing in groups of three, just like a three-letter word. Now, that's messenger RNA, and there's a start, AUG, and there's an ending point, UGA. So it's like a capital letter and a period. And in between that is the information that we've coded. So in the case of all three of these RNA molecules working together, one of the ones that is going to be very significant is called transfer RNA. And transfer RNA is going to bring down with it a amino acid. Now, there are only 20 amino acids. So we have a special code, and this particular one, of transfer RNA, on the bottom here has a sequence, a codon sequence of UAC. Now, we actually don't call it a codon sequence, we call it an anticodon because it can be complementary to the codon sequence on messenger RNA. For example, messenger RNA has a first sequence of AUG. The anticodon, or its complement, would have a sequence that you can see here, which is UAC. Now, if that's the right mix and it matches up, it will temporarily bond to this, the two together, and this amino acid that's right here is going to stay stationary right there until another transfer RNA molecule comes down, and if it has the right sequence, in this case, the U, the G, and the C, here's its complement. So if it's carrying with it another amino acid, what's going to occur is a temporary bond, not a temporary, a bond is going to occur between one amino acid and a second amino acid. This is a very important bond. It's called a peptide bond, and this peptide bond links the two amino acids together. Now watch this. As these are now attached, this tRNA is going to detach from its amino acid and go get another one. So what we're going to see here is a sequence of amino acids beginning to attach to one another. Once the transfer RNA has made its contributed its amino acid, it's going to go out and find another amino acid and bring it back down to that same ribosome. So it's a cyclical thing. So this will continue until all these amino acids are in place, one after another, in a particular order based on the code down below here on messenger RNA. So that's an extremely important event, and this is the assembly of amino acids in a particular order. So in my case, I have got, here's messenger RNA going all the way across, and it's describing a message from the first amino acid, which is like a capital letter, to the second amino acid, third amino acid, fourth amino acid, fifth amino acid, till finally it reaches like a period where that's my gene information. So really what we're getting at is a protein is a series of these amino acids linked one after another in a particular order with a type of bond called a peptide bond. This is our finished product. Now, it's not that simple, but we've left out a lot of the fluff, or I just include just the fluff to get you a starting point. So once this has all been assembled, then this protein will detach itself from the ribosome and then have the ability to go work within the cell to form a particular, maybe it's an enzyme that does a particular job or role. Maybe it's something like insulin that's produced in a cell, like in your pancreas, and will be released outside of the cell to have effects within blood sugar levels. So these proteins assembly are really the first part of what functionality this will do. But first of all, we have to make it. And we make it by that information that's stored on the DNA molecule. So in a sense, this is probably what we refer to as the simplest structure. We call it the primary structure, where we have a series of amino acids all linked together in a particular order. Now, what you'll notice here is that look on the left-hand side here where ASN is. This is an amino acid. It's just an abbreviation for that amino acid, but it's right next to one called GLY or glycine, and then phenylalanine, and then glutamine. So all these have names. I've just chosen to use an abbreviation of those. But the key element here is that they're linked together in a particular order and sequence. And eventually they'll form some more complex structures that we would later down the road call a protein. But this is the beginnings of that. So protein chains are made up of these amino acids, and that's really the key takeaway today. And the other part of that is that there are only 20 of these amino acids, and they can be arranged in a variety of ways. For example, if I took this yellow amino acid, I'll just say it's the color, and removed it or put it at the very end, that's a completely different protein. So order matters, and the order is written on those instructions, which is on DNA that we copied and then assembled based on that information on messenger RNA. So some of these proteins are building blocks, and they make up all the things that are you. In other words, some are very functional. Some are enzymes that break down milk sugar called lactase. Some are structural proteins that make up muscles. So all of them have a particular function, and they're based on a series of amino acids. So when we're looking at the order and sequence, it does matter. The use of messenger RNA codon chart is one way that we can interpret how amino acids are assembled. So the next two slides, I have, for example, how to interpret what codon sequence spells for a particular amino acid. For example, remember that we said that each three bases up on top here is referred to as a codon. So this is a codon chart. There are only 20 amino acids. So for example, if I had a amino acid sequence of a codon sequence of A, what amino acid does it code for? Well, what we have to do is use the chart, and the first letter here, which is A, means that we've designated that it's got to be somewhere in here. Well, the second letter is also an A. So this is my second letter up on top here. So if we go down where they all intersect, both the first and the second letters intersect, then somewhere in this box is the code for a particular amino acid. Now, it seems like there's two of them in here. For example, ASN has an AAU. ASN also has an amino acid sequence or codon sequence of AAC. Lysine, however, has what we're looking for. Lysine has a sequence of AAA, or it can have a sequence of AAG. Now, in this particular example, lysine was what we're after. And this is my third letter right here. So that's how you would use this chart to say, well, what does this mean, AAA? Well, it codes for lysine. And that would be, in this case, maybe our first amino acid. Now, the other thing to check out here is that there are a couple of these that code for something called a start codon sequence. For example, AUG is oftentimes like that capital letter that starts the sequence, and that's the methionine. And then there are a couple up here, UAG and UAA and UGA, that are like a period that stop the sequence. So there's a starting point and an ending point, and then all the rest of these are used to fill it in. And I know what you're thinking. You're thinking, well, how many of these are possible? Well, the smallest sequence of amino acids that I'm aware of is I think cow insulin has, like, for example, 64. 64 amino acids. That's the smallest one. So we're talking about an incredible number of amino acids linked together to manufacture one particular protein. This is another way of looking at it as well. This one is, I think, sometimes a little easier. The bigger the letter is the first sequence. So, for example, AAA, here is my first sequence. Here is my second sequence. And my third sequence right here, A. So A, going from the inside to the outside, codes for an amino acid called lysine. And again, just a different way of interpreting this. I think your book uses the previous chart, which is the square. So if we're looking at this, where we have a series of codons, for example, what's the difference between somebody that can taste a certain chemical called using PTC paper? If you taste it, I have it in class, it's really sour. Either you have the gene that you can detect it, or you don't. So for example, the taster, its sequence, if I just take a small section of it, one section is GGC, CGT, CAG. This is on my DNA molecule. Well, the non-taster, you'll notice that the C here has been replaced, or is replacing a G. And then it looks like the rest of this is pretty similar. Here's another difference right here, the A instead of a G, and then the T instead of the C. So there's a couple differences here. As a result of that, it codes for something a little bit different. For example, if this is my DNA molecule here, this is what messenger RNA would spell out. So for example, CCG, I'm wondering what amino acid that would be. I'd go over here and CCG, well, that would indicate an amino acid called proline. On the non-taster, at least the first sequence, here we have a CGC. Well, a messenger RNA would code for GCG. So here's GCG. Well, that codes for an entirely different amino acid. So this is a mutation that is prevalent here that either you can taste or you can't taste based upon the sequence of amino acids. So again, from start to finish, Translation is the finished product where you're taking the assembly, which is messenger RNA. Messenger RNA is located right here. You can see the sequence. It's attached to this blob of this gray or tan blob, which is my ribosome or rRNA. And then you can also see in action here my amino acids that are attached to transfer RNA. So this is where my transfer RNAs are going to come down. They're going to try to find a complementary base pair on my messenger RNA. And then if they do, they'll begin to link those amino acids together here. And this is where you can see the assembly begin to occur based on that. Again, once the transfer RNA has lost its amino acid, it's going to go out and get that same amino acid somewhere out here in the cytoplasm and bring it back down to again try to find another pair.